AS in A-level biology 9700, component number 11, May, June 2023. The electron micrograph show the onion root cell prepared using freeze fracture technique. In this freeze fracture technique, we are freezing first the cell or tissue, and then we separate different cells from each other. And during this separation process, the different part of the cell get damaged. So we need to identify from this diagram which organelles are being represented in structure X, or which part of the cell is visible in the structure X. So you can see if we are observing clearly, so this is cytoplasmic inclusions are visible out around. So in the center of the cytoplasm or inside of the cytoplasm, so internal, a largest part of the cell is visible. So obviously this is nucleus. And inside the nucleus is bounded by the double membrane. The outer membrane is visible, and the inner part of the membrane is also visible. So if you're seeing about the options, which options are given there? So number one option, which statement can explain the appearance of electron micrograph? So if you're observing about the electron micrograph, so the cell were broken apart, the endoplasmic reticulum, and the structure X is a ribosome, which is present on the endoplasmic reticulum. Here, there is no network of membranes are visible, which is formed by endoplasmic reticulum. So that's why it's completely smooth surface, no wavy appearance is visible. So that's why endoplasmic reticulum is a wrong option here. Second, the cell was broken apart at the nuclear envelope, and the structure X is a nuclear pore. So these nuclear pore are available. Why? Because these nuclear pore are visible on the outer membrane, which is facing to the cytoplasm, and inner membrane, which is facing to the nucleoplasm. So that's why these are nuclear pore. While the cell were broken apart in the nuclear envelope and the structure X is a ribosome, these cannot be ribosome. Why? Because ribosomes are not attached to the inner surface of the nuclear envelope facing to the nucleoplasm. So that's why structure X is not ribosome. Now, the cell was broken apart in the tonoplast, and structure X is a plasmodesmata. So plasmodesmata is basically a connection between the two cell, cytoplasmic connection between two cell. So in this way, the cell wall properly should be visible. So that's why these type of indications are not there. So that's why this D is wrong option. Question number 2 says, which cell structure can form vesicles? These vesicles are single membrane-bounded organelles, and these organelles sometimes contain cellular products, sometimes cellular excretion, or sometimes hydrolytic enzymes are present in that. For example, we can see the cell membrane, and inside of this cell, we have a rough endoplasmic reticulum, which is making protein, and protein is bounded in the vesicle. These vesicles are heading toward the Golgi bodies, and these Golgi bodies are making vesicle in a final packing, and these final packing are moving toward the cell membrane for exocytosis. While the cell membrane is also performing exocytosis or endocytosis. So these three structures, the endoplasmic reticulum, cell surface membrane, and Golgi bodies are making vesicles. So this is option A is correct for. For the student, were asked to match the function with the appearance of some cell structure in animal cell. So these students were asked the structure to match with the functions. For example, we can see the synthesis of polypeptide. So this is the function of ribosome, which are present on the surface of rough endoplasmic reticulum. So we're going to see that which structure is mating with the appearance of organelles. So non-membrane bounded spherical structure are ribosomes. While the synthesis of lipid is the smooth endoplasmic reticulum is responsible for that, and the membrane which surround and enclose inner cavity. So that one, this option is V is correct, and X is a packing of hydrolytic enzyme that will remain in the cell. So the hydrolytic enzyme, which are performing the internal or external cell digestion. So it is formed by Golgi bodies. So these are membrane bounded sac arranged as a flattened sacs. So these are stalks of the flattened membrane, which are arranged, pile up upon each other and making the Golgi apparatus. So this is suitable for Golgi apparatus. So we can see the V option is for smooth endoplasmic reticulum, while the W is non-membrane bounded spherical the structure. These are ribosome, and X is a double membrane bounded that are the interspersed with the pores. So there are pores are present in across the outer and inner surface of the membrane. This is nucleus. While the non-membrane bounded cylindrical structure, these are centrioles. While the Z is a membrane-bounded sac arranged as a flattened sac. These are Golgi bodies. So here, which option is going to match with? So we're going to see that, and this option, for example, the option A is correct. So number 1 is our W is matching with function. Number 2 is a V, and number 3 is with Z. For solving strategy, when you're reading the question number 1, 2, 3, these are their function. So directly you can write the name of the organelles, and then in the next, you can match the function of that organelle quickly, and then you can proceed for answering. Question number 4, which cell contain tonoplast? The tonoplast is basically a membrane which is present on the surface of the vacuole. So the tonoplast, for example, root hair cell contain a large vacuole. So that's why the tonoplast present in that. Companion cell, they contain large vacuole. So the tonoplast is present in that. And endodermis cell in the plant, they contain a large vacuole. So that's why the tonoplast is present in that. What about sieve tube element? They do not contain large vacuole or prominent nucleus. So that's why the tonoplast is not present in the sieve tube element. So D option is correct here. The question number 5, which organelles found in animal or plant cell are surrounded by double membrane? So when we see in the cell of a eukaryotic cell, so certain organelles which are surrounded with a double membrane, for example, chloroplast, mitochondria, and nucleus. While here, the wrong option is vacuole. Vacuole is single membrane bounded organelles, which is called tonoplast. So B is our correct option, chloroplast, mitochondria, and nucleus. Some scientists think that mitochondria evolved from bacteria, and they enter the cytoplasm of different cells and were able to survive there. Which structure feature of the mitochondria support this hypothesis? So the cellular organelles present in the cytoplasm, chloroplast, and mitochondria. These are double membrane bounded, have a circular DNA, and they have the 70 Svedberg ribosomes, as like bacteria. So according to the endosymbiote theory, these were the ancient bacteria who were engulfed by the eukaryotic cell, and they started to live in the cytoplasm of eukaryotic cell by symbiotic relationship. So that's why the folded internal structure is not is related with this hypothesis. So that's wrong. What about circular DNA and 70 Svedberg ribosome is related with this hypothesis. So these are two correct. So D is our correct option here. What is present in all viruses? all prokaryotes and all eukaryotes. So these some component of a DNA or RNA are present. For example, ribose, sugar, deoxyribose, cytosine, nitrogen base, and thymine nitrogen base. What about these all components are present in prokaryotes and eukaryotes? Why? Because prokaryotic cell and eukaryotic cell, they contain RNA and DNA. What about the viruses? These viruses contain either RNA or either DNA. So the ribose, some viruses contain DNA, so the ribose sugar is not present in that. They contain deoxyribose sugar. What about some viruses contain only RNA virus, or these are retrovirus? So they do not contain deoxyribose sugar. What about the thymine? The virus having RNA, they do not have the thymine nitrogen base because in RNA, thymine is replaced by uracil. So that's why the cytosine is the part of nitrogen space, which is present in RNA and the DNA both. So that's why all viruses, prokaryotes, and eukaryotes contain cytosine. Here it's the test of Benedict solution test. The table show the step that can be made to carry out the Benedict solution test. So which combination of these steps required to carry out semi-quantitative test? Basically, we classify the test into quantitative or qualitative results. So these semi-quantitative mean it's type of a qualitative test, but we are making this test as a quantitative by having standardized calculations. So to have standardized calculation, for example, the standardized volume of the Benedict solution and the volume of the test solutions. So if that the confirmed or measured volume of a Benedict solutions with the measured volume of the test solution we take, so this will be our semi-quantitative. What about the boil with the hydrochloric acid and neutralize with the alkali is not standardized. Why? Because volume of the hydrochloric acid and alkali is not mentioned. While the standardized boiling time with the Benedict solution, like how long we need to boil, so this is the semi-quantitative result. So that will be included in that. What about the second column is not standardized. So the definition for the semi-quantitative test is what? It's kind of the standardized test, which is placed in between two certain range, highest and lowest range. So the value will always come between these two range. So this type of test is considered as a semi-quantitative test. So C is our correct option here. The diagram show the three examples of different bonds. Now, first bond is a hydrogen bond, which is placed in between amino group and the carboxyl group. And second is our peptidic, and third is a disulfide bond. So which bond is held in the secondary structure of the protein? So secondary structure of the protein is maintained in the form of beta-plated sheet and alpha helix. So only the one component that's responsible, that is hydrogen bond. So that's why the C is our correct option here. So question number 10, the insulin is a globular protein which involved in a cell signaling. It is transported in the blood plasma from the cell that synthesizes in a target cell. So the molecule in insulin that contains a 6 sulfur-containing amino acid and has the 2 polypeptide chain. So which statement about insulin are correct? Now, here it's a mention like insulin is a globular protein. That's number one. Number two, if the insulin is containing the quaternary structure, mean 2 polypeptide chain are present in that. So if there are the 2 polypeptide chain are present, so we can represent, for example, these are 2 polypeptide chain. You need to be careful. They're showing this is our quaternary structure. And second, this is the globular protein. So if this is the quaternary structure, so an insulin molecule has a quaternary structure. This is our correct option. Second, an insulin polypeptide is held together by the 6 disulfide bonds. So 6 disulfide bonds only are not defining the quaternary structure. So that's why it is not a correct statement. So what about, there are few disulfide linkage are present inside of the insulin, but they are not describing the structure, quaternary structure of, or globular quaternary structure of the insulin. Now, amino acid with a hydrophobic interaction. So globular structure is designed by the 3-dimensional coiling of polypeptide in which hydrophobic bond or hydrophobic interaction is present inside of the globular protein, while hydrophilic is present on the surface. So hydrophilic have the same attractions around the molecule. So that's why it will get the globular structure. So our correct option is 1 and 3. C is our correct answer. Which statement correctly explains why the blood plasma can be maintained at the stable temperature? So the blood plasma can be maintained with the stable temperature. So why? Because it has high specific heat capacity. Mean it can absorb a lot of amount of the heat and change low temperature. So change less in temperature comparatively. So that's why it has a high specific heat capacity. Now, it has a low specific heat capacity. Is a wrong option. Why it is the wrong option? Because this option here, the low is wrong. So why? Because if it would be low, so it won't able to maintain its temperature. Now, it has a low latent heat of vaporization. So here the vaporization is not a relevant option. Why? Because the blood is enclosed into the blood vessel, so it is not vaporizing to maintain its temperature. So that's why B is our correct option here. The function of enzyme is to lower the activation energy. Now, this activation energy is a minimum energy which is required to convert the reactant into the product. So the graph shows the activation energy with enzyme and activation energy without enzyme. So which region on the graph shows the activation energy of enzyme catalyzed reactions? So we can see the function of enzyme is to lower the activation energy. So that's why the reaction is taken place at a low temperature, is catalyzed by the enzyme. So B is our correct option. So that's representing the enzyme catalyzed reaction. What about A is the difference in activation energy with enzyme and without enzyme. And C is the overall activation energy without enzyme. And the D is the difference of energy present in the product and the activation energy with enzyme. The graph shows the effect of an increasing substrate concentration on the rate of enzyme catalyzed reactions. Now, along x-axis, the substrate concentration is given there. Along y-axis, we have a rate of reaction is given there. Now here, the P is the reaction which is taking place at standard pH value and temperature. So there's a balanced pH value and temperature. So the question is, which description of change to the reaction condition could result into Q if all other conditions were kept same? So if we keep the other conditions same, so what kind of the one change took place which transfer this rate of reaction to decrease lead to Q? So what is responsible which lead to Q? So when we see this type of reactions, like this reaction initially it's same, and if you're increasing the actual substrate concentration, at the end, it get its normal condition. So this is our non-competitive enzyme inhibitor. So behavior of non-competitive enzyme inhibitor, so it is not affected by the substrate concentration. So when we see about non-competitive enzyme inhibitor, it is a wrong option. Why? Because it is not affected by substrate concentration. So aid inhibitor that attract to the site other than active site, that's a non-competitive inhibitor. So if you're increasing the substrate concentration, rate of reaction should not be affected by. So second option, we're going to check that. The second option is the aid, the inhibitor that is similar to the shape of the substrate. This is competitive inhibitor. Now, if you're increasing the actual substrate, so the rate of reaction will restore to the normal. So that's why the 2 is a correct option. Aid an inhibitor that block the active site of an enzyme. So if you're adding the inhibitor which is blocking the active site of enzyme, so because it is competitive inhibitor, it is correct statement. The number 4, the rate of reactions will be higher than to the normal. So if you're increasing the temperature, the rate of reaction will be higher. So when you're increasing the temperature, so we're going to see that the rate of reaction is lower. So that's why this is a wrong option. So if you're increasing the temperature at high temperature, the Q should be above the P. So that's why our correct option will be 2 and 3. That's a D is our correct option. Which statement about the cell signaling is correct? Now, cell signaling is a way of communication in which one cell communicates with another cell. So the cell is producing some kind of chemical. These chemicals are called stimulus, and these stimulus they combine with a specific receptor. Receptor will change its shape, and this will activate the G protein. G protein in turn activate the second messenger response, which are a group of enzymes present inside of the cell, and they release some chemical to activate one enzyme, which activates second enzyme, which activates third enzyme. This is called cascade with amplification reaction. And finally, the final enzyme is activate, which is responsible to produce a specific type of a change, and which is the correct response to that stimulus. This whole process is called cell signaling. Now here, we have the ligands or molecule which bind with a metal atom by a coordinate bond. This is the coordinate or dated bond, so in which one pair of electron is shared by the one atom to the metal. So here, which statement about cell signaling is correct? So option A, one type of receptor molecule will organize all the ligands in the body. So in this way, one type of the receptor molecule will recognize all the ligand in the body. So one type of the receptor can recognize more than one. Fine, it's possible, but not all. So the word all is basically wrong here. Second, the binding to the ligands may cause the change to the shape of the receptor. So the change to the shape of the receptor after binding to the stimulus, that's possible. In this way, the receptor will change its shape to activate the G protein. So this is correct statement. Now, the receptor for the ligands are always found on inside of the cell. So word always wrong. It may present above the surface or inside of the surface. Now, the same ligand is made by all the cell in the body. So the different cell, they are making the different type of ligands, or the different resonant molecule or resonant component of the ligand may change in a different cell. So that's why the word same is wrong. And here also all the cell is wrong. So whenever in the question and these type of the words are coming, for example, all and always and only or not, always be careful about that. These are the indication of answer. Here there's a question. The 4 students, A, B, C, D, observe the plant epidermal cell. They had been placed in a concentrated sucrose solution for 30 minutes. So the concentrated sucrose solution is an important point for you to identify the answer. They were asked to identify the partially permeable layer and explain the appearance of cell in the term of water potential and a movement of water molecule. So when we are observing about partially permeable membrane, so cell surface membrane is a partially permeable, while the cell wall is a fully permeable. But if there's a concentration of a concentrated solution, so the more water will move the inside to outside of the cell. And if there's a dilute solution, so the more water will move from outside to inside of the cell. So according to this concept, we're going to check that partially permeable membrane. So this is cell surface membrane. What about the cell wall? It's not partially permeable. It is fully permeable. So what about lignified cell wall is impermeable. So here, this is normal cell wall is being mentioned in the question. So that's why we can say cell wall is not partially permeable. So AB is our correct option. What about CD? We are not going to proceed with that to save time. Now, the second, the water potential at the start of an experiment. So if we place these tissue in a concentrated sucrose solution, so the water potential inside of the tissue should be higher. So that's why above option A is wrong, because it's saying the cell content have a lower water potential. So in this way, the B is our correct option. We just gonna verify this in the third column, the movement of the water during experiment, the water, more water moved out of the cell because it was placed in a concentrated sucrose solution. So B is our correct option here. The table compare the surface area to the volume ratio of the 5-agar block for the different dimensions, but which of all have the same volume. Now, all having the same volume, but their other dimensions are different. Now, here the concept is the rate of diffusion is directly proportional to surface area to the volume ratio. The object which has a more surface area to the volume ratio, the rate of diffusion will be greater. And the object which has a low surface area to the volume ratio, rate of diffusion will be low. So in this way, we can conclude. So the first option, it's saying the efficiency of diffusion will decrease as the width of the block increase. So when we see that the width of the block, so if you see that in option number 4, the width is 34. So if there's a width is 34, it is the highest value in this column of the width. So when we see that the surface area is 4.1. So that is the greatest surface area in this column. So in this way, the diffusion should increase instead of decreasing, because by increasing the width, the surface area is increasing. So that's why it is wrong option. While the B, they're saying the efficiency of diffusion will increase as the height of the block increase. So when we're gonna see that in the height, the highest height is what? 8 millimeter, and this is present in 1. But if you're increasing the height, so see that surface area to the volume ratio is decreasing. When there is a low surface area to the volume ratio, so rate of diffusion should decrease instead of increasing. So that's the wrong option here. While the option C, the efficiency of diffusion will increase as the block of fixed volume is flattened. The flattened mean if you're increasing the width of the substance. For example, when you're increasing the width and the length. So for example, if there's a width is 32 and the length is 32 and the height is 0.5 mean we minimize the height. So in this way, it became flattened. So in this way, the surface area to the volume ratio is 4.1 is the greatest. So that's why it is a correct option. While the D, the efficiency of diffusion will decrease as the block of the fixed volume is elongated. Elongated means if you're increasing the length. So you can see that if you're increasing the length is 64, so it's comparatively, it's a second highest value. For example, 1.5 is a second highest value in this surface area to the volume ratio. So by increasing the length, there should be a minor increase of the diffusion. So what about here in option is given decrease. So that's why it is wrong. So C is our correct answer. So we're going to proceed to the next question here. The cell cycle include mitosis. Which feature of this type of the nuclear division? So that mitosis is a type of asexual reproduction in which one cell is producing genetically identical type of daughter cell. So it is used in repair process, growth, and vegetative propagations. For example, number one, the cell of equal size to the parent. So the new cell, parent cell is producing two daughter cells, which are genetically identical, but they are smaller size. And after G1 phase, they will complete their normal size. So that's the wrong option. And that's form genetically identical. That is correct option. Semi-conservative replication of the DNA. Yes, it is a form of semi-conservative replication of the DNA, but this is not the part of M phase or nuclear division phase. This is the part of the interphase of the cell. So that's why the 3 is wrong option, because it's not relevant to nuclear division. So in this way, the only two is our correct options. A student observe the cell in a growing region of an onion root and obtain the data. So here, most important thing is observe. So what we are observing, so which percentage of the cell contain the chromosome that appear as 2-chromatid. So the chromosomes are only appear when the cell is dividing. So here, when the cell is in 2-chromatid, so the 2-chromatid are only present in G2 phase of interphase and prophase and metaphase. While in anaphase, these chromatid get separate from each other and start moving to the opposite pole. So that's why G2 phase, we are not including. So in G2 phase is a part of the interphase. So why we are not including? Because during interphase, the chromosomes are not visible. They are in the form of the chromatin material and they are not in the condensed stage. So that's why the interphase will not be included. While in the anaphase and the telophase, the chromatid will get separate from each other. The chromosome can only appear in prophase and metaphase in paired condition. So that's why for calculation of the percentage, the desired cell divided by the total number of cells multiplied into the 100. Now, total number of cells are what? 1,000. When we add them all together, like 886, 73, 16, 14, and 11, that is equal to 1,000 cell. Now, desired cell is equal to the prophase plus metaphase divided by, that's going to be 1,000. So this is what, and multiply into 100. So if you are going to divide it, for example, prophase and metaphase cell, so that's going to divide it by 1,000 and multiply into 100. So if you're going to multiply this cell into the 100, so that will give us a formula like percentage 73 plus 16 divided by 1,000 multiply into the 100, and this will give us the value 0.89. So multiply into 100. So if you're going to multiply into the 100, and this will give us 8.9%. So that's B is our correct option here. Which statement about the messenger RNA is correct? So statement about the messenger RNA, in a eukaryotic cell, messenger RNA is made by removing exon from the primary RNA transcript. So during the RNA, the transcriptions, so intron sequence are removed from exon sequence. So that's why this is wrong. Exon is wrong there. So this is called RNA splicing. The messenger RNA is a single-stranded polynucleotide containing a different purine bases. In a messenger RNA, in pyrimidine, the thymine is replaced into a uracil in messenger RNA. So that's why it is wrong. It is pyrimidine. Now, messenger RNA molecule contain ribose sugar joined to the phosphate group by phosphodiester bond. That is the correct statement. So there's a ribose sugar is present, and different ribose sugar are connected in a nucleotide by the phosphodiester bond, and it's making polynucleotide unit. Monomers of the messenger RNA contain phosphate group and deoxyribose sugar. So here there's a ribose sugar is present. So this is the wrong option. So here, the C is our correct option here. Which structure are involved in the transcriptions only? Now, in the transcription, so DNA template strain. So DNA is providing the template strain for the transcription. Fine, that's involved. What about RNA polymerase enzyme is transcribing the DNA to the messenger RNA that's involved in that transcription. What about anticodon as a part of the transfer RNA? And this is involved in translation process or translation of the protein. So that's why B is our correct option here. One gene provides the code for production of which type of the molecule? So gene is defined as a gene is a length of the DNA, which code for specific protein. So this protein is called polypeptide. So that's why the D is our correct option here. Now, this type of question, the tables show some messenger RNA code that code for certain amino acid. Now, these are code for certain amino acids. So in this way, what about in question, it's a given there, the DNA template strain has the base sequence are shown there. Now, these base sequence contain 6 triplet codon in the DNA. What you're supposed to do, what would be the change in amino acid be if the first base in the 5th DNA triple, in the 5th DNA triplet codon was substituted for A base. So we can see that, for example, when you see this is the DNA sequence and I separate by the different color triplet codon, number 1 is ACA, number 2 is GTA. In the same way, number 5 is GCA, guanine, cytosine, adenine. Now this guanine is replaced by A. This is substitution mutation. So this will become ACA. Now, we need to do what? We need to transfer this into a messenger RNA. So in messenger RNA, there's no thymine. Thymine is replaced by uracil. So in this way, you can replace into a messenger RNA. For example, we can make in a messenger RNA adenine to the uracil, and cytosine to the guanine, and adenine to the uracil. In the same way, we're going to make it the 4. So opposite of this one in a 4, this one, thymine, 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 and opposite will become adenine, adenine, adenine. So here, we can see that what is the difference. For example, in the DNA, before in the 5th position, there was guanine, cytosine, adenine. We change into adenine, cytosine, adenine. So what about in messenger RNA, what type of change will come? This will become guanine to cytosine and cytosine to guanine and adenine to uracil. So CGU has been replaced to UGU. So it means it's supposed to be CGU, arginine, and it has replaced to UGU. So now it is cysteine. So our correct option will be here. So we need to find out the correct option. The arginine has been replaced to cysteine. So in this type of the question, what first you're supposed to do, there is a gene is given there. So you need to convert this gene into a codon on messenger RNA. And then after finding this change, so in the rough place, find that in the table. And in the table, which is matching, for example, CGU is matching with the arginine and UGU is matching with the cysteine. So it means arginine was actual, but if the change took place, arginine has been replaced to cysteine. So we're going to move to the question number 23. Which structure contains a cytoplasm with mitochondria and nucleus? Now, xylem do not contain both of these because these are dead cell or there's no cytoplasm present in that. While the phloem sieve tube element do not contain nucleus. While the companion cell, these companion cell contain both the mitochondria and the nucleus as well. What is the correct term to describe the intermolecular hydrogen bonding between the water molecule? So intermolecular hydrogen bonding is what? The hydrogen bond is present between partially positive hydrogen atom to the partially negative oxygen atom of another molecule. And this force of attraction is called cohesive forces. And this is called cohesions. So B is our correct option here. What about adhesion? So this is the force of attraction of water molecule to the lignified cell wall of xylem, that is cohesions. while osmosis is not related with diffusion, and osmosis is the transport for, so the water molecule move from a high to the low concentrations. So by osmosis, and it's also diffused. Here, the diagrams show the outline of the xerophytic leaf that has been left for 45 minutes in a different condition of P and Q. So we took the plant leaf and we placed it in a P and Q, two different solutions. And we are observing in the solution P that the part of the leaf become turgid. So it means this solution was dilute solution, hypertonic environment. While in the Q, the part of the leaf become plasmolized or shrinked. So it means it is a hypertonic solution, or it is concentrated solution or hypertonic environment. So which statement is correct about the P and Q after 45 minutes? So there is a less negative water potential in P than in Q. So if there is a less negative water potential, mean there is a high water potential. So that's why we can say, like in a P environment, there is a high water concentration because it is dilute solution. So if there's a high water concentration, so the water move from outside to the inside and it will make the cell turgid. So this is the correct statement. The cell may be turgid in P and plasmolized in Q. So in P, they are the turgid and they are plasmolized is visible because it's shrinking. So that is the correct statement. The reason is what? The P is present in dilute solution and Q is present in concentrated solution. So now the option, the cell are less turgid in P than Q. But in actually, the cell are more turgid in P as compared to Q. Why? Because water is moving from outside to the inside more in P as compared to the Q. There is no net diffusion of water into Y in either P and Q. So net diffusion means the same amount of water moving from outside to the inside or inside to the outside. What about there's a different amount of water is moving into the P, while different amount of water is moving outside. So more water is coming inside in a P, less water is moving outside, inside in Q. So that's why there's no net movement or equal concentration of the movement is there. So that's correct statement. So 1, 2, and 4 are our correct statement here. How does the sucrose move from the chloroplast to the phloem? So it means the sucrose, how the sucrose is moving from the source cell to the phloem sieve tube element. So here, it is moving from a high to the low concentration. So first, the sucrose dissolves into the water, and that's moved from a high to the low concentration by diffusion. And then it moves inside of the cell wall. So that is apoplastic pathway. And then from cell wall, then it will enter into the companion cell, and from companion cell to the sieve tube element through the facilitated diffusion. So that's why this is the cell cytoplasm to the cell cytoplasmic movement from companion cell to the sieve tube element by plasmodesmata. It is a correct symplastic pathway. So these three pathways are followed by the sucrose to moving from source cell to sieve tube element. How about the companion cell involved in loading sucrose into phloem sieve tube element? So how it is responsible for loading the sucrose? So the sieve tube, first the companion cell, they pump hydrogen ion or proton outside of the cell into cell wall to increase the concentration of hydrogen ion outside. And this hydrogen ion start moving from outside into the companion cell. And the same way, the sucrose follow the same protein channel as a co-transport. So that's why the sucrose, which is moving into the companion cell by the co-transport, and these protein channels are open, which are driven by active transport of hydrogen ion. So first actively through the co-transport protein, and then passively through plasmodesmata. So passively plasmodesmata mean the sucrose transfer from the companion cell to sieve tube element. What about actively through the co-transport and the protein and the plasmodesmata? What about the plasmodesmata transport is not active transport. It is driven by facilitated diffusion, while passively through the co-transport. So the co-transport is generated or is driven by active transport, or actively through the plasmodesmata. That's the wrong option. So A is our correct option here. The photomicrograph show the blood cell are seen using the high power light microscope. Now, these blood cell, number one is a neutrophil. Why? Because it's comparatively large with a multi-lobed nucleus. So there's a multi-lobe nucleus, meaning many lobes are there in the nucleus. This is visibility. So number one sign is it's supposed to be a large, number one. Number two, it has a one nucleus with contain many lobes. So that is neutrophil. Second, there is a compact structure. Monocyte is the largest in a compact structure. What about the phagocyte? They have no the specific shape, and this contains a specific compact shape. And it has the two-lobed nucleus. It's a large two-lobed nucleus, so that is monocyte. While the lymphocyte are comparatively smaller cell in white blood cell, and they contain a dense, large proportion of the nucleus. So that's present. So these are lymphocytes. So you need to identify the number one is the neutrophil, number two is our monocyte, and number three is lymphocyte. Here, that's the correct option. Now, the question number 29, the plant diagram of the two cell vessels are shown there. Now, these the two blood vessels are given there. So these blood vessels are, number one is artery. Why? Because there's a narrow lumen and it contains comparatively thick walls. So what about second one? It contains a large lumen with a thin wall, and that is the vein. Now, in this vein, the outermost these are fatty tissues, and second are connecting tissues or external. So they are the external adventitia or connective tissues or ectoderms. So these are collagen fibers. Below the ectoderms, we have the mesoderm, and this one is the layer. This contain the smooth muscles, and these smooth muscles are thicker in artery and comparatively thin in the vein, while the endothelium that's present in both. So here we need to find about the correct option, and the correct option will be number one is our artery, number two is vein, and number three is our collagen fiber, while the number four is our smooth muscle, the next layer, and the five, the both is present, the endothelium that is present in both. What about other option? They are varying these part. So in this way, you can see the B and C are the relevant correct option. And then further, you can proceed to the next from B and C. So 3 is collagen fiber in both, while 4, there's elastic fiber are given. So that's wrong. So in this way, you can proceed to the next for finding the correct option. So our correct option is here, B. Now, next question is, what are the found in the blood and tissue fluid? So what is present in the blood and tissue fluid? So the carbon dioxide, this is the metabolic waste. This is present in both. Fatty acid, these are nutrient after digestions of lipids or byproduct of metabolism. This is present in both. White blood cell, they provide immunity. So that's present in the body tissue and tissue fluid and the blood. And next we have protein. So the protein that is in a form of hormones or insulin and glucagon or antibodies or blood clotting factor like albumin, fibrinogen. So that's present in, these all are present in tissue fluid and the blood. The 31, which reactions would be slowed down by the inhibitor of carbonic anhydrase? So we're going to see that which reactions are directly involved which carbonic anhydrase is directly involved in which type of reactions. For example, carbon dioxide react with a hemoglobin and it's producing carb aminohemoglobin. It's kind of van der Waals force in which the enzyme is not involved in that. Second, the carbon dioxide react with the water and produce hydrogen carbonate. So this reaction is catalyzed by carbonic anhydrase enzyme. Now, if there's a more number of carbonic acid will produce, there is more dissociation will take place. So if the less number of carbonic acid will produce, less dissociation will take place. So that's why number 2 and 3 directly depend upon the carbonic anhydrase. So they will be directly affected by inhibitor. So D, the 2 and 3 are correct option here. We're going to see that there is the sequence of blood flow is mentioned. So the blood flow in the heart. So in a cardiac cycle, so we have a sinoatrial node, which will give electric supply to the right and left atria. And there will be contractions of atria, right and left atria, and blood will transfer from atrium to the ventricle. And then atrioventricular node will receive the electric supply from sinoatrial node because the sinoatrial node are connected with the atrioventricular node only in the ventricle part. Now, in this way, the atrioventricular node will give electric supply, and this will give excitation and send electricity to the Purkinje tissue. These electrical impulses move to the Purkinje tissue, and in this Purkinje tissue will cause a contraction in ventricle wall, and the blood will move from ventricle to the outside. So that's the sequence. So which option is following this sequence? That is the correct option. For example, number 1, X. So this X is what the wave of excitation spread from sinoatrial node. And after that, if there's a sinoatrial node, it's giving excitation. So number 2 is the T. Atrial wall should contract. And then the V, the wave of excitation enter into atrioventricular node. And then U, it will pass for a second. And then W, the wave of excitation pass to the Purkinje tissue. And after that, number 6, so that's the Y. And there will be the ventricle contractions. The blood will move from the heart to outside. So here we're going to proceed to the next. The number 33, the diagram gives the information about the blood pressure and the left side of the heart during the cardiac cycle. So when we're observing about the blood cycle, so it's easy to left side. Why? Because we can observe the pulse rate, the blood coming from aorta. So here we can see there are different parts where the different valves are opening and closing. For opening and closing of wall, if there is a high blood pressure at front of the wall will close. If there's a high blood pressure at the back of the wall, so this valve will open. So in this way, when we can see this one, at the point number 1, where there is an atrioventricular wall, which is present between atria and ventricle, is closing. Why? You can see that the ventricle pressure line, there's a line which is ventricle, there's a dotted line you can see. So this line is increasing as compared to the atrium line. So it means the pressure in ventricle is increasing as compared to the atria, so the atrioventricular wall will close. Now, this way, the ventricle contraction is start and the ventricle pressure increase as compared to the aortic pressure, aorta. And when you can see this dotted line pressure is increasing as compared to the aorta. So in this way, the high pressure at the back of semilunar wall, so this will cause the semilunar wall will open. But the blood, as the semilunar wall will open, blood will constantly transfer from ventricle to aorta. So that's why the both pressure is constantly increasing. But the time will come when the volume of blood start decreasing in ventricle. So in this way, the ventricle pressure will become less than aorta pressure. So in this way, the semilunar wall will have a high pressure at front as compared to the back side. So in this way, to prevent the backflow, semilunar wall will close. And constantly, the blood pressure will decrease in ventricle, and the time will come, the atrium pressure will be greater as compared to the ventricle. And in this way, the atrioventricular wall will once again open. So in this way, there's a point number one, atrioventricular wall will close. Point number two, semilunar wall will open. And then point number three, the semilunar wall closes. And four, atrioventricular wall will open. So we're going to see this one according to this information, what is the correct option. So here, an atrioventricular wall will open. This is our point number four. While the semilunar wall will open, this is the two. Atrioventricular wall will close, that's a one. And the semilunar wall will close, this is three. So D is our correct option here. We're going to proceed to the next question here. A person with no breathing condition rests for an hour. So in this way, if the person there's no fluctuation in the breathing and it's a breathing with a normal rate at the time of rest. So the person's heart rate is also remain constant. So which statement is correct? So the carbon dioxide concentration in the blood in pulmonary vein will be higher than in the pulmonary artery. So this word higher is wrong. So the pulmonary vein contain deoxygenated blood, which is coming from the heart. So the carbon dioxide concentration in the blood in pulmonary vein will have higher concentration of the carbon dioxide because it's moving to the lungs, and in the lungs it will remove the carbon dioxide. So that's why this concentration of the carbon dioxide concentration in the blood in the pulmonary vein. So this is, the carbon dioxide concentration in the blood in the pulmonary artery is greater. Why? Because the pulmonary artery is getting blood from the right side of the heart, from right ventricle, and that is deoxygenated blood. So this deoxygenated blood has a high concentration of the carbon dioxide, and then it will move to the lungs, remove the carbon dioxide, move to the pulmonary vein. This contains a high concentration of oxygen. So the word higher is wrong. The pulmonary artery has lower the concentration of the carbon dioxide than pulmonary artery. The option B, the carbon dioxide molecule in the air of alveoli move out of the blood by active transport. The gases in the plant and an animal anywhere cannot move by active transport. So that's why word active transport is wrong. The gases always move one place to another place by simple diffusion. The next is the air in the alveoli has lower concentration of the oxygen. So comparatively, when we are observing about the concentration of the air in the air, so this is oxygen in the air is a 104 millimeter per mercury. What about the oxygen is in the blood? This is 100 millimeter per mercury. So always the partial pressure of oxygen in the alveoli in air, so it will stay higher. So that's why the word lower is wrong. The option D, the oxygen molecule diffuse from air into the alveoli, into the blood at a slow rate, and then to the person is inactive. So if the person is not active or in a sleeping mode, so the rate of diffusion will be low. Why? Because the tidal volume is a 500 milliliter cube only. So that's why if there's a tidal volume is 500 milliliter cube out of 5,000, you're breathing in and breathing out, the rate of diffusion will be less. The which statement about all the bronchiole are correct? So the bronchiole produce after the bronchus. So the trachea divide into bronchus, bronchus divide into bronchiole. So these bronchiole, they have epithelium, fine, it's correct. They have muscle, they have the smooth muscles, which control the diameter of the bronchiole and provide flexibility. While they have goblet cell, there's no goblet cell are present in bronchiole. So that's why option 1 and 3, the B is our correct option. The next question is about the diagram show some of the pathogen that cause disease in a human and some of the way they are transmitted. So these pathogen, which are causing the disease and their pattern of the transmission is given there. Now question is, what is the correct pathogen and the method of transmission for malaria? So malaria is caused by Plasmodium. and the plasmodium is related with the kingdom Protoctist. So that's why the number 2 is our correct option. That's Protoctist pathogen, while it is transferred by a vector animal, that is mosquito. So mosquito is responsible to transfer the plasmodium from infected person to the normal person. So that's why the X is our correct option. So C is our correct option here, 2 and X. Some of the process which result in formation of the population of bacteria that has resistance to the new antibiotic are listed. So what is the process? How the bacteria get resistance to the specific type of antibiotic. So we need to arrange them in a sequence. So this sequence is given first, we have a random mutation, which produce a different type of alleles. And in this way, if the different type of alternative form of genes are produced, so genetic diversity come in the populations. So it means it increases the different type of alleles in the populations. Now, in these different type of allele, some are advantageous, some are disadvantageous. So in this way, some are the more favorable, some are less favorable. So the more favorable are called advantageous character. So these advantageous allele have reproductive success. Meaning if they have more survival, they can reproduce more. So if they can reproduce more, so it means these resistance allele will increase in number in the population. Why? Because if they have more survival, more reproduction, more number of organisms. So that's why this sequence we're going to follow, the sequence will become first is a 4, a random mutation, and then the increase in genetic diversity, and then number 1, change in reproductive success of the bacteria, meaning it can reproduce more. And number 2, that increase in frequency of resistance allele. So that's D is our correct option here. The second last question, these are the 38 question is that before second last, when a bacteria are grown in a petri dish containing disc with antibiotic, there will be a zone of the inhibitions of a bacteria. So if you're growing the bacteria and you're going to put the certain type of antibiotic, it will kill the bacteria. So if the antibiotic is more effective, so this inhibition circle will be larger. And if it is less effective, this circle will be small. So this way, we're going to see that the size of this circle is represented by this bar graph. So zone of inhibition along y-axis, while the type of antibiotic along x-axis. So we're going to see that the most effective antibiotic is number 3, while the least effective antibiotic is number 2. So the question is about the type of antibiotic, which is the most effective, or type of antibiotic, which is the least effective. So you can see the most effective antibiotic is number 3. Why? Because it's producing a greater amount. So what about a greater or larger size of inhibition, while the least effective is number 2. That's making a smaller size of inhibitions. Now, second last question here, which blood type does not recognize, engulf, digest the non-self particles? So there's a type of blood, this is what T killer cell. T killer cell can recognize the non-self antigen on the body cell, which are infected by the non-self antigen, but they're not involved in engulfing and digestion of the non-self particle. So that's why they can recognize, and they recognize these non-self particle on the surface of body cell. They come in contact with the body cell and release cytotoxin, which block the metabolism of the cell, and they kill the cell. So which are infected by non-self particle, but they are not involved in engulfing and digestion as like macrophage, neutrophil, and phagocytes. So D is our correct option here. The repeated infections with the malaria result in the more effective immunity to the malaria. So which type of immunity is responsible for the more effective immunity? So if we are suffering again and again by malaria, so malaria contain different type of pathogen. So these different type of pathogen, if they are attacking on our body, so there is enough time for our immunity system to produce multiple different type of antibodies about different variety or strain of malaria. So in this way, this type of natural active immunity will produce enough and sufficient amount of memory cell, which can produce the enough or sufficient amount of plasma cell to produce plenty of antibody to give a quickest response of the second entry. So that's why the natural active immunity is considered most effective immunity. What about the artificial active immunity is also playing its role, but the number of the memory cells are comparatively less. Artificial passive immunity is a temporary based immunity, and natural passive the immunity is also a temporary based immunity, while the long lasting, permanent, and more efficient immunity is a natural active immunity. Here we have completed. Thank you very much for staying with a long time for completion of this paper. If you don't understand any question, so you can also write in the comment box and I will explain individually that question for you. Thank you. Thank you very much.